Okay, so let's look at immunoglobulins here. Immunoglobulins is the fancy way. We already know what these are. You've heard them before. Immunoglobulins is the fancy word for antibodies. And you'll see it shorthanded as IG, okay? And immunoglobulins are proteins, okay? They are physical structures. They go out and they hunt for a very specific thing. They look for a specific type of antigen. Antigen are the surface markers, and they let us know that we are we or you are a foreign invader to you. Okay, so antigens are what are going to tell the body that we are us or a foreign invader, and it's the antibody's job to go out there and basically tag those things. They tag them, they stick to them, and that's basically a beacon to say, come here and kill this thing. Antibodies are there for viruses, they're there for bacteria, toxins, yeast spores, anything really. So they're really good. They are soluble, and humoral immunity is what's going to be the process involved with helping to destroy all of these, excuse me, foreign invaders. When you look at its structure, and I believe the next slide has a picture of its structure, it's comprised of 4 polypeptide chains in the shape of a Y, basically. Okay. Okay. That's basically kind of what it looks like. All right. 2 identical heavy chains, and then 2 lighter chains. Okay. A couple heavy, a couple light. It's more of the functional stuff that we'll probably get into a little bit more. Variable and a constant region. Okay, so of the 2, that's what we're probably going to get into a little bit more right here. So immunoglobulins or antibodies, proteins in the shape of a Y that go and attack foreign invaders or a foreign antigen. So there you go. There's the Y-shaped structure of the immunoglobulin, the antibody. Okay, and we have a variable region, and we have a constant region. Okay, and here's the variable region. Okay, it's at the end of the arms. Okay, and this is where we have the antigen binding site. Okay, and antibodies usually have 2. So there you go. This is where this antibody, immunoglobulin, goes out and binds to the foreign invader. It does it through some weak intermolecular forces there, basically, some hydrogen bonds or just some weak type of bonding. That's all it does. So here at the variable region, that's where we bind to the foreign invader. Then we have a constant region, okay? And you can see it's all of this stuff right here. Okay. All this constant region. And this constant region determines what its true function is going to be. Does it attack parasites? Does it attack bacteria first? Does it attack viruses first? All right. So the variable region is what varies. It changes depending upon what the antigen markers are and binds to it. The constant region basically tells us its function. Okay, it's biological function. And there's 5 different classes of them, and we're going to have to know kind of what they specialize in. But they are IgG, IgM, IgA, IgD, and lastly, IgE. Those are our 5 types of antibodies, our 5 types of immunoglobulins. Very simply, this is what antibodies do. Okay, they bind. When they bind, that's when the magic happens. All right, they're going to undergo neutralization, agglutination, and precipitation. These are the 3 major actions. 1, 2, 3. Neutralization, agglutination, and precipitation. These are our 3 main functions for antibodies. Okay. Neutralization. We have to neutralize this foreign invader. We have to neutralize whatever is Virus, I believe, is on this picture. Yes, virus is here. Okay. Neutralize the foreign invader, virus, bacteria, whatever the case may be. Okay. So to do that, we have to cover it. Ultimately, this is going to, if we cover this foreign invader, we're going to be attaching to this thing, and we are not going to allow it to bind to a cell, to enter a cell, and get our cells to do its bidding. Okay, so it neutralizes it. It neutralizes it and makes it ineffective in establishing an infection. It makes it ineffective in getting into the cell. We've neutralized it. Then we do this thing called agglutination, clumping. Okay, it clumps. We are sticking together. It forms a big clump. This does a multiple number of things, as you might imagine. It prevents it from just floating around willy-nilly, but it kind of gets them all to kind of stick and clump together there. And so we can focus our attack from all of our other immune system components that we've been talking about. As you can kind of see, it's especially effective against bacterial cells, but whatever. It's all antibodies are going to do these three things. And then precipitation. First step for an antibody, bind to it, cover it. Make it impossible for that foreign invader, virus, bacteria, whatever, to be able to get into a cell. So now it can incorporate its genetic material into the cell's genetic material and get the cell to do its bidding. Second step, we've neutralized it. Second step, let's clump it up. Let's agglutination. Okay, let's clump it up. This prevents them from spreading all over the place, getting into tissues, getting into various different structures, prevents the excessive spread of the individual foreign invaders. Then we precipitate. These antigen, antibody, cross-linking things, all the little secretions that go with viruses as well, we get it to become insoluble, okay? And this allows it to precipitate out of solution, right? That's the terminology. If it was soluble, it would mix in with the plasma, right? With the water component of blood. This antigen-antibody complex that happens as soon as the antibody binds to the foreign invader, it starts to come out of solution. And when it comes out of solution, now I've clumped it all together. We're coming out of solution. So it's a big structure there, and we can get all of our various phagocytic entities within the immune system to come there and start chomping away at it. And we get rid of the infection. Obviously, this can take a little time. The first time you see it, or the second time you see it, the first time you see a foreign invader, all of this takes a lot of time. The second time you see it, much, much faster response. But all antibody as IgG through the IgE, all of them do these three things. Cover the surface, neutralize it, get it to clump together, and then get it to precipitate out of solution so I can chomp, and destroy it. What else do antibodies do? We have the variable region, which will attach itself to the various different antigen markers on the surface. Okay. And we have a constant region, and a constant region is always the same. Okay. It always does the same thing. It's going to be of the same class. So when we look at antibodies from a constant region, they'll do a handful of more things. It can allow for complement fixation. Complement fixation, these are going to be various different structures associated with helping the immune process out. They complement the immune system. You get an antibody that binds to it, it will release some chemicals, and it'll be the release of those chemicals that will activate this kind of complement system where it helps aid the process along. Okay. And here you can see there's the antigen. There I've bound to it there, and it helps to activate complement. It goes there and it helps to enhance the immune response. All right. And again, that's because of that constant region. IgE is always this. IgG is always this. IgA is always this. Opsonization. Okay. Opsonization. Opsonization, guys, is the ability for an antibody, immunoglobulin, to basically mark the foreign invader for death. That's probably the most simplistic way to think about it. So we have neutralization, where we attach to all of the foreign antigens on that surface, right, and prevent it from getting into a cell, get it to clump, glutenate, and all of this is done on that variable region end. And then we get it to precipitate out a solution, and then we can destroy it, chew it up. The opposite end of the immunoglobulin, the constant region, it can do a number of things as well. First one is it can call other things to the party to try to help the process out, the process of destruction of this foreign invader. And that's what complement is. It complements, it facilitates, it helps the immune response. Opsonization is the way of a constant region to mark the foreign invader for death. It basically gets a number of phagocytic type cells to come in there and see it and engulf it. So it can do it from the variable region end, and it can do it from the constant region end as well. And something else it can do, mark it for death. It'll mark it for death for a specific type of immunoglobulin. If it's bacteria, it's going to be this. If it's viral, it's going to be this. If it's a parasitic infection, it's going to be this mark for death type of thing. The last thing that antibodies will do, so there's 6 things. The last one is going to be activation of natural killer cells. Natural killer cells do a number of different things, as we've seen. One of the things they go is they're immune surveillance, right? We saw that earlier. And it is that immune surveillance, they're kind of this general kind of kill-all. Kills all defective cells, cancer cells like we talked about. But it will also release some cytotoxins that can get there and penetrate that plasma membrane of that foreign invader. And here you can see the natural killer cell. Here's the antibodies. Got receptors for natural killer cells here. Natural killer cells come in here, and they release a number of chemicals. Can release a number of chemicals that can perforate that foreign invader and kill it. Okay, and kill it. And as you can see, this is a virus that's gotten into the infected cell here, or into a cell, and obviously has infected it. And so these natural killer cells can not only aid in destroying the foreign invader, but it's also killing the cell, because the cell is not doing what it's supposed to do. It's doing the virus's bidding and not our own bidding. So this is a way to kind of get rid of the foreign invader, but also now this defective cell, and the cells can divide and we can replenish. So 6 different types of functions. Makes a great matching question type of question on an exam. Neutralization, agglutination, and precipitation. Not only that, we have complement fixation, opsonization, and activation of natural killer cells. So these are our 6 major functions. And as you can see, all of them are involved in the destruction of the foreign invader. It just goes about it by a number of various different actions. When we look at the immunoglobulins, guys, here they all are. IgG, IgM, IgA, IgD, and lastly, IgE. Here's our first ones here, IgG and IgM. IgG is the most abundant antibody found in blood. It's like upwards of 80%, somewhere in that ballpark. So it's the vast majority, okay? Most abundant antibody in blood, okay? In blood. It's also kind of in some other fluids like lymphatic fluid. So blood and lymph, interstitial fluid, you know, right? They're all kind of similar in components. But IgG is going to be that most abundant. It can also cross the placenta, so it can actually confer immunity to the fetus inside as long as the fetus has a placenta and an umbilical cord attachment to mom. Mom can confer immunity to her unborn child just through sheer IgG. It can participate in all types of antibody actions. It's actually going to be greatest involvement is going to be when you see the foreign invader the second time, so second exposure. Second exposure is going to be much more active. So anytime you're seeing it for the second time, IgG is going to be your man. IgM is found in blood as well. It's going to be most effective when you see it for the first time. More active in first exposure. Okay. Where IgG is going to be more active in the second exposure, IgM is going to be your first guy or girl to the party when it first sees a foreign invader. So when first exposure to a foreign invader, IgM is going to be the primary thing involved here. Found in blood, just like IgG. So these two are kind of your biggies when it comes to blood, okay? This one can cross the placenta. It can cause hemolytic disease of the newborn. That's where we have an RH incompatibility or an ABO incompatibility between the mom and the fetus. ABO incompatibility with the fetus is not very common, but it can. It's more of an RH incompatibility between mom and fetus. So IgG is kind of involved with that, or one of the things involved that will attack. IgM, again, when it first gets exposed, IgM is going to be the person on the scene. Again, it's also responsible for, mismatched or rejection of transfusions. Most effective to clump and to bind complement. Where it's found, when it's active, what it's responsible for, those are probably more important things to remember. The next two are IgA and IgD. IgA, as you can kind of see here, is predominantly found in all of your secretions. Mucus, saliva, tears, breast milk, okay? IgA is going to be the immunoglobulin that's found primarily here. It helps protect, what do all of these have? All of these secretions are open to the outside world, right? So in your areas where you're going to have exposure to the environment itself, IgA is pretty much always going to be associated with that immunoglobulin. So it helps protect the respiratory, the GI. That's where you see a lot of these things. Also, something you see here is breast milk. So not only are we giving immunity, or you, mom, are giving immunity to your unborn child through IgG, you're also giving them through IgA as well. Breast milk. Breast milk is rich in immunoglobulins. That's the one major difference between breast milk and And formula is the ability to have antibodies in it. So we can confer immunity, or you can confer immunity, women, to your newborn just by breastfeeding. So a pretty important aspect there. As soon as you cut the cord, no IgG. If you don't breast milk, or as soon as you stop breastfeeding, okay, or if you never breastfed, I guess you never had it, you're getting some immunity. So it's important, breastfeeding. And come to nutrition class, we'll talk about that there. Since it does protect the respiratory and the GI tract, it helps to prevent pathogens from adhering to the epithelium there and causing an immune event. So it prevents them from adhering and ultimately penetrating in the epithelium, anything that's exposed to the outside world. IgD is the last one on this slide here. And IgD is kind of interesting. They know some functions. They know, exactly what it does. It helps to basically enhance B cell activity, B lymphocytes. It functions, as you can see here, as an B lymphocyte receptor. They know what it does. It basically blows the trumpet to call in more antibodies and more immune cells to the foreign invader. Some of the exact how it does that, they don't exactly fully understand yet, but they do know what it does. And IgD is basically, it's the guy blowing the horn, calling in the cavalry. It enhances all of your B lymphocyte activity, and so it causes more, antibodies to come to the fight, and basically all immune components to come to the site of infection. The last one here is IgE. IgE is usually formed in response to parasitic infections or allergic reactions, okay? So, if you do give blood and you are fighting something off, or even if, you go to the hospital with some kind of weird infection that they can't just figure out, they'll do a complete, blood, a complete, CBC, differential, all that stuff. And so understanding what these immunoglobulins do, what all these antibodies do, and when they're more prone to be higher, can give you some real insight into figuring out how to approach the patient, how to treat the patient, or what to treat the patient with. So that's usually the only two things that IgE is elevated in. If you have allergies, you're going to have elevated, or if you have a parasitic infection. Usually this has a very low rate of synthesis. But when you do have an allergic reaction, it goes up through the roof. When you have parasitic infection, it goes up through the roof. What does it do? Well, basophils and mast cells come into the region there. Tracks eosinophils. Eosinophils were elevated with parasitic infections. Hopefully you can remember that one as well.